viewers, since last many weeks we are studying electron transport chain, and day by day or video by video we are covering complex 1, complex 2, complex 3, complex 4 of this chain. But still we are just one step to the formation of ATP. If you remember, in the last video we have covered the formation of water or the reduction of molecular oxygen. So the two major roles of electron transport chain, which are production of water and generation of ATP synthesis, and so far we have completed the one conclusion or the one role of this electron transport chain, that is to reduce molecular oxygen, correct? So before moving ahead to the complex 5 or the last complex of this chain, few things are left over and we will discuss these few things first, then we will move towards the complex 5 or the last complex of this chain. Okay, so oxygen, it is a very dangerous instance. Why so? Because wherever oxygen is there is a chance of formation of free radicals. Complex 4 having an ability to bind with molecular oxygen proceeds with the formation of water or end up to the formation of water, but the transfer of electrons one by one to the molecular oxygen can be serious and it can impose damage to the cells. How? Let's discuss that. These are termed as the side reactions on complex 4, or simply the formation of reactive oxygen species, or simply ROS. The ideal electron acceptor on this overall chain is your molecular oxygen, which reduces to 2 molecules of water. We know that this is the primary role of it, but partial reduction of oxygen can create hazardous compounds, and these are superoxide anion, peroxide and hydroxyl radical. Electrons, whenever they are single, whenever these are unpaired, are extremely dangerous, and this can easily happen when one electron transfer is going on, correct? So during the process, if you remember the last overall reaction mechanism on complex 4, there is formation of superoxide anion, but generally the structure of complex 4 or the prosthetic groups in this complex are arranged so beautifully that this superoxide anion, if formed, cannot escape out from the complex, so it normally cannot cause damage to the cells because of the structure and complexity of the prosthetic groups of complex 4. But in some cases, if these ions release to the cell, these cause harmful or dangerous side effects, and these are known as reactive oxygen species. So what if ROS formed? Correct? Normally it will not form or scavenged, but if it is formed, then the enzymes we have our army in the cells, and these are superoxide diasmutase and catalase. These two army which are present inside our cells scavenge these unwanted substrates to remove them, and they are the best examples of catalytically perfect enzymes. Have you heard this term before? Yes. Where? I want you to write in the comment section. We have discussed one more example of catalytically perfect enzyme throughout this journey. Okay, I'm not talking about electron transport chain, rather you need to recall from my previous videos. Okay, let's discuss what superoxide dismutase do. Okay, these are two superoxide anions formed. If these are formed and released, then superoxide dismutase scavenge them to form oxygen or molecular oxygen plus H2O2 or hydrogen peroxide by consuming two protons. Protons are very much empty in our inner membrane space. There is no problem of proton inside our inner membrane space. So if they are formed and released, superoxide dismutase scavenge them and combined with hydrogen ions to form two less harmful or less harmful products, okay, or the safe products, safe byproducts. Okay, now the role of peroxidase. Yes, there is another enzyme known as peroxidase. It also helps in this process in that if these two are formed, it utilizes oxidation of NADH2 by combining reduction of H2O2 to water. The valency or the oxidation state of oxygen in H2O2 is -1, but in H2O it is -2. So the basic need of the electron transport chain is to form water before before we go to the synthesis of ATP. So if superoxide is formed, yes, superoxide dismutase comes and convert it into oxygen in H2O2, but there is requirement of formation of water which is accomplished by another enzyme known as peroxidase. And there is catalase also, which also combined with the H2O2 to this byproducts to form water and molecular oxygen. So we are now safe. If free radicals is formed, we are now safe because we have three army which are very active and catalytically perfect. Means these are waiting for their agents or for their criminals to come to trap them and to form safe byproducts. Okay. Now, what about complex 2? We have discussed complex 1, complex 3 and complex 4, but we have not discussed about complex 2. So what is the role of complex 2 in this overall process is that it just accepts electrons from FADH2 which is formed in the citric acid cycle or in the mitochondrial matrix. We have not discussed this complex before because it is not involved in the pumping of protons. There is no pumping of protons through this complex, so we have not discussed it previously. Correct? It just accepts electrons from FADH2 and give it to Q or coenzyme Q, same as complex 1. Correct? Okay. So we are now at the complex 5. So our wait is over and finally we are at the complex 5 waiting for our flame or the body energy or ATP. Correct? The cellular flame or the ATP will be synthesized here on this ATP synthase complex. But there is a very interesting theory behind this. Till 1960s, there is no idea about how ATP is synthesized by this electron transport chain. However, scientists and researchers know that obviously there is something on this chain which is involved in the synthesis of ATP. But what that thing is or what is the theory behind it, nobody knows. Okay, so we know that the electron transport chain always couples the redox and phosphorylation reactions. Redox means reduction and oxidation. Phosphorylation means, obviously this thing you have not come across so far, because this will happen on this ATP synthase complex. The phosphorylation of adenosine diphosphate into adenosine triphosphate, or the flame of cells. Okay. So every time when you encounter ETC, you must remember that spontaneous electron transfers are coupled with non-spontaneous pumping of protons into intermembrane space. Correct? You should memorize this line because this is the key point of this overall respiratory chain. Okay. So try to just speak it as many times as you are encountering with this ETC. Okay, this is the best thing to remember. This is speak up. Okay. Now, so we were talking about theory. So it was known that the membrane potential, people know about the membrane potential, that the cellular membranes are charged itself. Okay. So a very strong, it was hypothesized that a very strong chemical gradient which is established with few protons in the matrix and many in the intermembrane space means how a gradient is formed. Obviously, protons are being taken up from the matrix and delivered to the inner membrane space. So there is less positive inside and more positive outside. So there is a ionic gradient formed across the inner mitochondrial membrane. Okay, it is a role of proton, or it is a beauty of proton which confers to the formation of membrane gradient. We know that. Okay. So generation of a pH gradient or the proton gradient and a charge difference, obviously negative in the matrix as compared to the inner membrane space, constitute a proton motive force that can be used to drive ATP synthesis and transport processes. Okay. So what I just want to explain through this slide is that it is the hypothesis of the renowned scientist Peter Michell, who was working in Cambridge University, England in 1960s, right? So it was his theory or his hypothesis that the proton gradient is generated with energy from electron transport by pumping of protons by complexes 1, 3 and 4. And the direction of proton pumping is from the matrix side to the inner membrane side of the mitochondria. What Peter Michell hypothesized is that, the another thing which he hypothesizes, that there is oxidation and phosphorylation, both these processes going on the electron transport chain. Yes, they had an idea of the electron transport chain. The Peter Michell, which had successfully completed his PhD on penicillin, working in the Cambridge University in the biochemistry department. Now he was working on this chain with a group of scientists, their post-PhD work or working as a research associate over there. So there's an interesting story is that the hypothesis, they were working on this chain, and there was no theory about how actually this ATP is synthesized. So the start of any scientific proof or evidence is that they first hypothesize, then they prove their hypothesis, and that results in the formation of a theory. Correct? So Dr. Hans Krebs has given Krebs cycle before that. So they have an idea about what is happening in the citric acid cycle. Okay, they have an idea about the membrane gradient also, that the membranes are charged itself. But how ATP synthesized, it was a vague idea. Some people were saying that It was conformational changes in the proteins which are present on the electron transport chain, and this conformational change is responsible for the formation of ATP. But only Michel, it was only Michel who was very sure that not any conformation, rather it is the proton motive force or the pumping of protons, it is actually involved in the synthesis of ATP. And the second thing which he knows, that the inner mitochondrial membrane is impermeable or not permeable to the protons, correct? So there are two things. The inner membrane is impermeable to protons, but protons are being pumped out. It means there is presence of one or many proteins on the inner membrane of mitochondria, which causes pumping of protons. Michel also hypothesized that the proteins which are involved in the pumping of electrons, which is known by the name of complexes, and the complex or the protein which is responsible for the formation of ATP, these two are separate entities. Please be focused on what I am saying, that the complexes which are involved in the pumping of protons are different, and the complex which is involved in the synthesis of ATP is different. But one thing is very sure, that they all are located on the same inner mitochondrial membrane. Correct? It was the hypothesis of Peter Michel. He was very sure in the hypothesis, and as soon as he delivers this hypothesis in front of others, he was hugely or largely criticized. So he has formed a separate research institute to prove that his hypothesis is actually right. And in proving this overall hypothesis, one of his friends named Moyle, Jennifer Moyle, he supported him in this overall journey or in this overall study. Okay, so what the things or what the prerequisites which are required to prove this scientific theory? The first question. Second thing which, or the other hypothesis which he gives, is that the protons have a thermodynamic tendency to return back to the matrix, and the term proton motive force was given to this tendency, that initially the protons have tendency to move to the both sides, from matrix to the inner membrane space, and down gradient from inner membrane space to the matrix, and this tendency to return to the matrix is called as proton motive force, and this is actually responsible for the synthesis of ATP, because free energy releases in this overall process. How? If you remember, in the last video I told you that one proton is equals to some amount of free energy. So pushing proton from lower concentration to higher concentration takes energy. So coming back of protons from the higher concentration to the lower concentration releases that much amount of free energy, and that drives the synthesis of ATP. It is very much clear. Now the thing which is unclear is that how to prove his hypothesis. Okay. So his hypothesis was proved by generation or formation of an artificial membrane or artificial lipid bilayer, which is termed as which is termed as synthetic vesicle. Correct? It is shown in this picture. The synthetic vesicle, the circular one, is artificially formed, and this actually represents or this exactly represents the lipid bilayer structure of cell membranes. Okay. Now, as Peter Michel says that hypothesized that proton pump is required because the membrane itself is not permeable for protons. So, for the proton pumping, one bacterial rhodopsin was used or was clubbed with this vesicle, and this was obtained from bacteria. Bacterial rhodopsin is present in some halogenic bacteria, as far as I remember, purple bacteria, yes, purple bacteria, and from that it was isolated and just combined with this vesicle. Okay. So, two things were required as per Peter Michel. One was the proton pump, and second is the ATP synthase itself, because ATP cannot be formed without this synthase complex. Okay, and the inner mitochondrial membrane was recreated by this synthetic vesicles. So, we have all three things together at one place. Now, what did he do? He has put this vesicle into a high proton concentration outside. Okay, the proton is present outside this vesicle, and this bacteriorhodopsin has ability to pump proton as soon as it comes in contact with light. So, a light source was put near this experimental setup, and as soon as light was turned on, the pumping of protons from the outside to the inside takes place, and this is what measured by the pH. It is very easy to measure the pH at one point to the other. So, it was clear that due to the measurement of pH, it was clear that yes, inside concentration of protons becomes high, inside becomes acidic. So, it was proof that first thing, that the pumping of proton is done, correct, pumping of proton is done. Second thing, this ATP synthase has taken this proton, pull out to the outside, and via this coming out of the proton through this complex, in the presence of ADP and inorganic phosphate, ATP or cellular flame is formed, correct. So, it was not just a 1 day or 1 year experiment. It took several years, because there was not much team work into this. Michel was alone, along with one of his friends. He has procured this bacteriorhodopsin from somewhere. ATP synthase comes from the chloroplast. Okay. So, what he proved, that only presence of proton pump and ATP synthase can lead to the formation of ATP. But in biological system, the pumping of proton is related with the transfer of electrons. So, if electrons are not there, you can just artificially synthesize ATP by having just a proton pump and a light source and your ATP synthase complex. And after this experiment, what Michel gets, not just ATP but also a Nobel Prize. So, we should be very much thankful to him and his colleagues for our current understanding of synthesis of ATP. He has devoted his life proving this theory, correct? Okay. This key experiment clearly showed that the respiratory chain and ATP synthase are biochemically separate systems. This is a key point. The respiratory chain means the overall electron transport chain having the complexes. These complexes are involved in electron transfer and also proton pump, correct? And ATP synthase is a separate entity. You can separate it out. You can still form ATP by just having a proton pump, correct? So, this is the finding and the conclusion of his theory to prove that the role of respiratory chain, or the ETC was played by bacteriorhodopsin, and this is how the story of formation of ATP comes to an end, and his work was finally recognized and he received a Nobel Prize. So this is the end of this lecture, and in the next video we will study about the structural details of this complex and the overall process of the generation of ATP. Till then, happy learning.